Good day, great dolls, and as you guys can see, today is the 25th of December, Christmas public holiday, but I am continuing to just quickly push out content so that come Jan, you guys have a lot of resources to refer to, and yeah, wishing you guys a happy Christmas, and yeah, hoping you guys are safe and you guys are actually now resting a bit and spending time with family. That being said, God bless all of you. Okay, let's quickly carry on. So, guys, today we'll be looking at DNA code of life, and this is the second section. And in the second segment, you'll see that we'll continue touching on the importance of DNA itself, and we'll also touch on aspects of RNA, and we'll be comparing the two towards the end. So let's quickly start. So boys and girls, we've found out in the previous lesson, if you look at the previous video, the first lesson, we found out that in DNA, adenine bonds to thymine and guanine bonds to cytosine. And we remember this by using the rhyme apples and trees. So basically adenine binding to thymine, cars and garages, cytosine bonding to guanine. And we said, now, why is this sequence, the way in which they pair to each other, important? Now, it is important because it provides the code which gives the instruction for protein synthesis. Now, boys and girls, when we synthesize something, we make something. So protein synthesis, it's important because it gives the code which gives the instruction for making proteins in smaller, or I guess in easier terms, okay? Then when we move along, we find out that DNA has functions, but now we want you to know specific functions, and these are all in alignment with the exam guidelines. So these are the ones that you have to know. So name the two functions of DNA. The first one is that DNA contains coded information for protein synthesis, and then the second one is that DNA makes up genes which carry hereditary information. So these are two important facts that you guys have to then know for the exam that's coming up. And then, boys and girls, we have a process called DNA replication. And when we replicate something, we're making an identical copy of that specific thing. So what is DNA replication, boys and girls? Now, boys and girls, DNA replication is a process whereby DNA makes an exact or identical copy of itself. We are making an exact or identical copy of itself. So what is DNA replication? It's a process whereby DNA makes an exact or an identical copy of itself. Now, you have to know the process. And in exams, the process is a good 6 or 7 marks. So it's very important for you to know the process. And this process is exactly according to what's stated in your exam guidelines. So don't go learn what Tani Suzanne from, I don't know what school, or Tani's, I don't know, whoever taught you what. But you have to know these things according to the exam guidelines because they're very strict when marking the work. So let's quickly go through the process of DNA replication, boys and girls. So as we know, DNA is double-stranded, right, boys and girls? So the first step is that the DNA double helix unwinds. So it unwinds, and then the weak hydrogen bonds between the DNA nitrogenous bases break. So when these bases break that hold the nitrogenous bases together, the two strands then unzip. Now, each of the original strands will serve as a template onto which its complement is built. Remember, DNA is a double-stranded molecule. So when it breaks up, one strand will serve as a template, and the other strand will serve as a template onto which its complement will be built. But how is this complement built? This complement is built by the free-floating DNA nucleotides in your nucleoplasm build a DNA strand onto each of the two strands of DNA by attaching them to the complementary nitrogenous bases. This will thus then result in two identical molecules being formed, and each molecule will have one original strand and the other one will have an old strand. So essentially what happens is that we have a double helix, it unwinds, the bonds that hold the nitrogenous bases together break, right? And when these strands break, right, boys and girls, what happens is that each of these strands will serve as a template so that we form another DNA. We are doing DNA replication. So we start as one DNA molecule, will end as two identical DNA molecules, right? So each of the strands that have been broken up serve as templates. Now, what happens is that free-floating nucleotides, boys and girls, in the nucleoplasm, right? What they do is they'll build a DNA strand by attaching the original strand, right, or the template, to its complementary nitrogenous bases. And as such, this will result in two identical molecules being formed, and each molecule consists of one original strand and an old strand, okay, and a new strand, I mean. Now, when does this take place? Now, this is a question that most people tend to get wrong. DNA replication, boys and girls, takes place during interphase. And where does it take place? It takes place in the nucleus. When? In interphase. Before everything. So it takes place when? Interphase. Where? Nucleus. Alright. It's very important for us to then look at the structure, boys and girls, of a chromosome. What is a chromosome? If you can quickly recall from the last session that we had. From the last session that we had, boys and girls, we did say that a chromosome is a long thread-like structure that comprises of DNA wrapped around proteins called histones. And a chromosome is very important because it contains the genetic material of the cell, and it contains basically the genetic material of the cell, which is hereditary. One second, boys and girls. Just give me 1 second because someone has decided to make a monster noise now. Okay, there we go. So, boys and girls, I hope I did not lock. Okay, cancel. There we go. Apologies, boys and girls. I just had to close the window because, as you know, festivities. So, we said that this is a chromosome, boys and girls. Okay. Now, I want us to know, why is this whole process of DNA replication significant? Why is it important? Why do we have to learn about it? What significance does it hold? Now, during mitosis, if you remember PMAT, mitosis is a process whereby we make identical copies of a cell. Okay? So now, during mitosis for cell growth and repair. And then also the second importance is that it doubles the genetic material so that it can be shared between the resulting daughter cells during cell division. So it doubles the genetic material so that it can be shared equally and normally between the resulting daughter cells. If this does not happen, we usually have what we call aneuploidy, whereby we will have one cell having more or less number of chromosomes. But we'll see that as we move along, boys and girls. I apologize. Now, I did talk about this. Now, where does DNA replication take place? It takes place in the nucleus. When? During interphase. Okay? It's very important for us to know this. Now, boys and girls, you guys will encounter a DNA profile. And in most cases, a DNA profile will be used when matching a suspect to, or linking a suspect to a crime scene, or when you're busy with paternity disputes, right? Now, what is a DNA profile? Now, this is the definition that they want you guys to know. A DNA profile is a pattern of a bars coincides, right, together with the sequence of base pairs. Okay? And we'll do past papers where we'll deal with a DNA profile, and then I'll teach you guys how to answer questions related to a DNA profile so that we get maximal marks. Okay? Now, name 2 uses of DNA profiling. I want you to know these 3 because they might give you one in an example, and they might say, except for the one mentioned in this investigative question when looking at an investigative question. The first one is to identify dead bodies. The second one, which I said, was paternity disputes, where you don't know who the father of your child is, or you are, I guess, having doubts about being the father of someone. Hopefully that never happens to you. And then from there on, matching tissues for organ donating. That's where we use DNA profiling. Now, name a view. They can ask you to name a view, especially in these investigative questions. Name a view against DNA profiling. Well, we know that we're humans, so human error, boys and girls, can occur. Alrighty. Now, boys and girls, we are now moving on to another molecule. This molecule is called RNA. Now, as we said, where does DNA occur? We said DNA occurs in the cytoplasm, it occurs in chloroplast, and in the nucleus. Now, where does RNA occur? RNA occurs in the nucleus, in the cytoplasm, as well as where? In the ribosomes. Ribonucleic acids, ribosomes. 2 and 2 is 4. Makes sense? Makes sense. Okay. Now we're going to move on. And boys and girls, DNA has a nucleotide. RNA has a nucleotide. And when we think back, we said that a nucleotide is the monomer or building block of RNA and DNA. So now let's look at an RNA nucleotide, boys and girls. An RNA nucleotide consists of 3 things: phosphate group, a sugar molecule. Now, what sugar molecule would RNA consist of? RNA would consist of what? Ribosugar. Ribosugar, okay? Now, from there on, we'll see something very funny that tends to happen. I want you to think about this scenario that I'm going to give you as we move along. You'll see as time goes. So now, RNA has nitrogenous bases. But in RNA, adenine does not bind to thymine. Adenine binds to uracil. And cytosine binds to guanine. So cytosine and guanine are like in a committed relationship. They're like married. They don't cheat on each other. They don't do funny things, which you'll never find in this day and age. But with adenine, adenine binds to uracil. So I want you to remember this. So adenine switches up on you and denies knowing you the same way your partner denies being with you in front of his or her friends, eh? Adenine switches up when seeing its friend RNA and says that it's in a relationship with uracil. So adenine is 9 out of 10 times like your boyfriend or your girlfriend switching up on you. But in anyways, that's your problem. You're still the stupid one. But anyways, I'm just joking, guys. Let's carry on. So please remember it that way. So in DNA, adenine binds to thymine. In RNA, adenine binds to uracil. Alrighty. So now when we look at the structure of an RNA nucleotide, we can see it's very similar to that of a DNA nucleotide. It's just that we will here have a ribosugar, we'll have the same phosphate group, and then what changes here is that instead of having thymine, we'll have what we call uracil. So here, the nitrogenous bases for RNA are adenine, uracil, guanine, and cytosine. Okay. Now, let's look at the shape of RNA, boys and girls. We said that the shape of DNA in the first scenario was double helical. It was a double helix shape. The shape of RNA is single-stranded. So therefore, there are no hydrogen bonds. It's just a single strand, okay? Now, let's quickly move on to talking about the different types of RNA that we encounter. It is very important for you to know what I'm saying, and these notes that I've written down and that I'm going through and I'm explaining to you. As the viewer, it's very important for you to either screenshot these and know them off by heart, because when I hate shouting now, and I hate dealing with people that act dumb and are stupid. By the time you're learning this, learn these notes. You won't need another textbook. You won't need a the answer series, because some of the answers I've made notes from the answer series. I've made notes from the other textbooks that are issued by the by the Department of Education. So do me a favor, know these notes and then just do past papers. You will be fine. You should be obtaining nothing less than an 80%, because if you are obtaining anything less than an 80%, then I don't know what's going on. I failed at my job. Okay. That being said, I'm calming down. I'm no longer crashing out. Let's carry on. So, now it says name 3 types of RNA. We have mRNA, which is messenger RNA. We have tRNA, which is transfer RNA. And we have rRNA, which is ribosomal RNA. And I want you to take note of the way in which I've written these M, T, and R's. They're in small letters, and RNA is in caps. Should you dare think about writing everything in capital letters, I will give you a slap. Your teacher can just give you a 0 there, because that is not the way in which I've taught you stuff, and that is definitely not the way in which they've taught you the work. So don't be stupid. Do as we say, and you will succeed. Okay. Now, functions of RNA, boys and girls. We said, what is the function of mRNA, which is messenger RNA? Messenger RNA, you're carrying the message, the genetic code. Okay. So messenger RNA or mRNA, it carries the genetic code from DNA in the nucleus to the ribosomes in the cytoplasm. The function of tRNA is that tRNA picks up specific amino acids. tRNA, transfer RNA, it picks up specific amino acids in the cytoplasm as per the coding of the anticodon bases and takes them to the ribosomes for protein synthesis. rRNA plays a role in protein synthesis. Now, boys and girls, these are very important for you to know so that when we move on to the third lesson and we're learning two different processes, transcription and translation, you'll see how easy it moves. DNA replication, you must know because it's gonna also link in with transcription and translation. The functions of different types of RNA, you must know they're gonna link in to what? To transcription and translation during protein synthesis. Very important for you to know. If you know this, that process will be a breeze for you. Okay. Now, boys and girls, you encounter terms like codon. Now, what is a codon, boys and girls? A codon is 3 consecutive nitrogenous bases on DNA, and an anticodon is 3 consecutive nitrogenous bases on RNA. Codon, DNA, anticodon, RNA. Okay. Now, I want us to quickly just look at this because this tends to pop up and it's usually in your investigative questions also. It will be a good 5 marks, 1 mark for actually having a table and 4 marks for the content. Okay. Now, let's look at the 3 differences between DNA and RNA, boys and girls. These you should know. DNA is double helix or double helical shaped. RNA is single-stranded. Now, DNA consists of what we call deoxyribose sugar, and RNA consists of ribose sugar. Now, DNA also consists of thymine being present and uracil being absent. RNA consists of thymine being absent and uracil being present. Now, boys and girls, people are deciding to be very stupid again and are deciding to play loud music because I don't know, for some odd reason they're saying it's December. But lucky for me and you, I'm done with presenting lesson 2 to you guys. And that being said, boys and girls, may the good Lord bless you. May he keep you and may make his face shine upon you as you continue studying. Boys and girls, thank you very much for supporting me. Thank you very much for subscribing and liking. It truly does mean a lot to me. I don't think you guys know this, but yeah, it truly does mean a lot to me. And I pray that, you know what, your work, your hard work, and what I guess you reap what you sow. So your hard work will not be in vain. I can promise you that. Okay. So remember to take care of yourselves. If no one's told you this today, you are loved, and you are special. You are unique, and you can make it, and you will make it. Okay. All the best, guys. Please drop your comments in your questions or your comments or your remarks in the comment section. Thank you very much, guys, and God bless.